So I'm Rob Howes from Charles River. I'm one of the co-track chairs. Kelly Gray is the other co-track chair from Bionevix. And I'll just give you a couple of minutes of intro, not to the track, but actually more to the sustainability work group. So I'm also I'm on the ELRIG board. I head up the sustainability work group, and this track has kind of come directly out of that work group. So within that work group, we're really trying to look at what can we do to make both ELRIG as an organization, but also as a community more sustainable. So we're looking at a lot of things, both in terms of the venues for the big conferences, how we make that as sustainable as it can be, especially being a conference organization, there's a lot of travel involved. But also then looking around the rest of the community, what are the things that people are doing across our various organizations that are really good examples of increased sustainability? And then how can we make sure that we share that information across all of the ELRIG community, but then start to implement that as well, so that we start to see substantial benefits. So we're a fairly new group. We've only been going just over a year. We are actively looking for other members. So if anyone is interested, contact myself or Kelly or any of the other work group members. If you want to know who's on the work group, we're all listed on the ELRIG website, so you can find a list of us there. We're a fairly diverse bunch, but we'd certainly be looking out for other members. So that's enough about the work group. Let's get into the science. So I'm going to hand over to Kelly.
So welcome everybody. Really excited to obviously see a sustainability track in the main auditorium for 2025, and hopefully this will long continue into the future. I hope you'll agree that we've put together quite a diverse and broad programme, which hopefully is something for everybody in there thinking about how to embark on their sustainability journey. Some things will be new, some things will be obvious, and some things will be not so interesting. But hopefully there's something that you can take away from the meeting. What we're really hoping for is that there's some light bulb moments go off. So you can really think, I can apply that in my work, or I can think about how to do that. And that would be a massive plus from the meeting today. So I'm going to stop talking now, because that's the best thing that can happen really. And I'm going to introduce our first speaker. And this is Ross Kent, and he's the Global Manager of Field Application Scientists at Synthace. And he leads efforts to integrate sustainable automated design of experiments, so DOE, in the drug discovery process. So Ross has been a passionate supporter of using DOE in biological research and uses his own first-hand experience to help others apply the methodology to their lab work. He educates scientists across the industry and then works closely with them to implement DOE and automation in existing research projects and programs. Ross has got a diverse background in molecular biology with experience both in industry and academic R&D. He's held positions at Syngenta, where he worked as an automation scientist, and at the University of Manchester and the Royal Dutch Shell in Houston, Texas. He earned his PhD in synthetic biology from the University of Manchester, focusing on the application of DOE and machine learning to the optimization and design of gene expression circuits. So I'd like to take the opportunity to welcome Ross to the stage, and we can listen to his talk.
Cool. Thank you very much for the kind introduction, Kelly. It's a pleasure to speak with you all today. As we've said, I'm going to be talking about how we can use the Synthace platform to ultimately drive smarter experiments and hopefully achieve some greener outcomes. So I'm actually going to be sharing this talk today with a co-presenter here, Harry Hickey. Harry's joining me from GSK, where he's going to be talking about how they've been implementing Synthace within their own assay development labs in Stevenage. So hopefully this isn't too of a controversial slide, but I think we all kind of know that drug discovery can have a little bit of a sustainability problem, right? The processes that we do are incredibly resource intensive, whether that's plasticware, tubes, tips, energy for running incubators and freezers. These things take a lot of carbon, a lot of resource to do. There's also then the chemical waste that comes out the back of a lot of these processes. And I'm not going to go into too much detail on this, and there are some talks later on that are going to touch on it a bit more. And ultimately, then I think really as an industry, the biggest challenge we face is the sunk carbon cost and energy investment we make into drugs that ultimately don't actually make it to our patients. This is a challenge, right? And it's something that, I think we can all agree we need to try to improve, not just for financial reasons or for the cost benefit, but ultimately for sustainability reasons as well. So, little bit of a blip there. So those are sort of obviously some industry level challenges that we all kind of face. But ultimately where this can kind of, the rubber meets the road, if you like, can be with a scientist in a lab with a pipette in their hand, a load of tubes and an experiment that they have to run, right? Now, as biologists, we're often having to juggle a lot of things when you're in the lab, whether that's thinking about the protocol you've got to execute, the instructions, the samples, the data, analyzing that data, and then reporting it at the end. So, but Synthace, we kind of have this view that that's a lot of mental burden, that's a lot of things that people are having to think about day to day. And asking those scientists to then add on to that, well, how do I perform this experiment in the most resource efficient way is, it's really a lot to ask. So just I guess, a little bit of a diversion here, just to tell those of you who maybe don't know too much about Synthace, what it is that we do. So Synthace is a cloud-based platform that ultimately allows you to digitize the entire experimental process. And this is from initially designing and planning your experiment. I'll show you the sort of interfaces we use to do this in a second. To then running that experiment in the lab. So you can describe your intent, this is what I want to do with some liquids. These are how I want to mix some cells together. We then take that plan, generate some instructions for either liquid handlers or for manual execution for you to then go in the lab and actually run the thing, right? We can then take the data, structure that for you, give you some tools to then analyze it, fit some models, and ultimately plan your next iteration through this cycle. And what that looks like is generally, this is sort of the heart of the Synthace platform at the moment. It's what we call a workflow builder, and it allows you to just break down your plan into a set of different steps. So we have these elements, they might be used for making mixtures, diluting something, and you can use them to, as I say, describe what it is you're wanting to do. You then click this little button in the top-hand corner here. Synthace will look at your plan, look at the devices you've selected, and generate the instructions for you to run. You then get this nice preview, and this allows you to just visualize all of the liquid transfers that are going to happen. But I guess from a sustainability perspective as well, will give you information about all of the resources that you're going to require to actually execute this experiment. So that's all the tips, tubes, reagents that you're going to need to actually go away and run this in the lab. So we kind of see Synthace fitting into sustainability in two sort of key ways. The first is that resource information that we're providing, and I'll touch on that in a little bit more detail in a second. But then secondly, because we have that digital representation of your experiment, we can really then provide very complex experimental designs to that model of your process, if you like. We use this to allow scientists to implement DOE or design of experiments in their process optimization and development. And don't worry, I will talk about DOE in a bit more detail. Surprise, surprise, a Synthace talk about DOE, I know. So on that first point, this was a nice paper that we published recently in partnership with UCL, University College London, where they were basically looking at what does it practically take to implement automation in a lab? And they did this quite simply. So they took a very simple process where they're just culturing some cells in some antibiotic media. They looked at executing this manually and in an automated fashion, and asked then 4 or 5 different scientists from different sort of levels of experience to go away and take this protocol and run it however they see fit. I think they gave it to an undergrad, master's student, postdoc, and a PhD student, and just said, right, tell us how you do it. They then sort of observed these key three key things. They looked at the time taken, the number of errors that were made during the execution of this protocol, and then the plastic waste that was generated. And what they found was really quite interesting, maybe not that surprising. Depending on who you give that protocol to, they're going to execute it in different ways, right? And what they found was that there was a lot of variability in how efficiently people worked in terms of that plastic waste that's generated. Nicely, when they then did things through Synthace, they found they were actually able to, by standardizing that protocol and having it digitized, they're able to reduce the variability in how much waste people were generating. The second point is that because they then have that model of that experiment, you can very easily and rapidly go back to your workflow, make some modifications to, let's say, how tips are being reused or the labware that you're using, and really sort of quickly iterate and minimize the resources that are being used in that process. And I'm not going to read this all out, but let's just say, I guess our marketing team were delighted to hear this, that, you know, Synthace ultimately allows users to evaluate their consumable requirements before actually setting foot in the lab, right? So you can ask questions like, oh, how do I modify this process a little bit to cut down on the tips that I'm using, let's say. So that's kind of on that first point about providing that upfront resource information. The second point really on DOE. So I think it's fair to say that biological research is pretty hard to do. I think we all know that in this room. And drug discovery can be pretty wasteful. And by that, I mean, you know, if we look at failure rates of drugs later on in the development process. And at Synthace, we kind of have this belief, if you like, and hopefully Harry can prove this out for me later, that part of the reason that we face these challenges is that the experimental methodologies that we use to ask questions of biological systems is ultimately insufficient, right? So typically as scientists, and you know, we're all taught this in primary school, the way that you experiment is to fix everything, change one factor at a time. So to take an example here, let's say I want to understand how temperature and pH impact a system. I might vary temperature across this axis here, run those 5 experiments, find my optimal temperature, and then start to vary pH. But the reality here is that I haven't really fully explored or understood that little box here, that 2-dimensional space, and I'm ultimately missing things. I have a flawed understanding of the system that I'm working within. What we actually find is that instead of our optimum being here where we initially thought it is, it's over here somewhere. Now, one solution to that is to just say, well, I'll just test every single temperature and pH combination, right? That is impractical, incredibly resource intensive, and ultimately very expensive to do, even with just 2 factors. As we start to add factors into this or variables that we're wanting to explore, that obviously explodes very quickly into impossible to run experiments. This is where DOE comes in. So the eagle-eyed amongst you might have seen in the workflow builder earlier that we have this little toggle here where we can say, right, I want to take my plan, I want to apply DOE to it. I won't go into this in too much detail. Drop by the booth if you do want to hear more about it. But just to sort of outline what DOE ultimately is, it's a tool that allows you to systematically sample experimental spaces. So you can select a sort of strategic set of runs, go away, run those designed experiments, collect some data from them, and then use some modeling to basically fill in the experiments that you didn't do. So that way you can get that understanding that you need, but in a very efficient way. And then we get some nice predictions from that model and say, oh, actually, I think you want to be operating over here somewhere rather than in the middle here. So to very briefly, in a high level, sort of summarize the benefits of DOE when it comes to sustainability, DOE is ultimately about driving efficiency, but without compromise, right? So we can test multiple things at once. I can test my little example here, temperature and pH. So 2 hypotheses for 1 experiment, that's great. You can obviously you can push that much higher. We have customers and users who will do 20, 30 factor experiments at once. That's 30 hypotheses that you're testing in one go. You can also drive efficiency, as I say. You know, DOE can allow you to be very run efficient in the work that you do. It's really about doing less work, getting more information, and gaining some cost and sustainability benefits along the way. So ultimately, you might all remember the reduce, reuse, recycle triangle from primary school. I certainly do. DOE is all about just reduce what you do less, but make it count more. And at Synthace, we have this belief that one of the key areas that I guess we've seen be successful, and Harry's going to talk to in a minute, is in assay development. So assays are the decision-making tools that we all use, right? They are the foundation of a lot of what we do in the lab. With high-quality assays come high-quality data, and ultimately with that comes gains in both short-term and long-term efficiency. But you don't just have to take my word for it. I'm going to hand over to Harry at this point, and he's going to talk a little bit about some of the work they've done at GSK to implement DOE and some of the sustainability gains that they've received. So.
Thanks, Ross. So I've got 3 examples of where we've used DOE and it's improved the sustainability of some of our processes, okay? So the first example is based around IMIDs. So IMIDs are small molecules which bind to cereblon and lead to the degradation of neosubstrates. And this can have tetragenic effects. I'm sure many of you will know about thalidomide, which is one of the IMIDs. So because of this effect, initially, most of our cereblon binders within GSK were classified as high risks. We note that as OHC5, which is an OEL of 1 or lower. And so because of this classification, it comes with extra steps that have to be involved in the handling process. So double gloving, double bagging of waste, weighing the compounds in a separate hood to the others. And then when we get rid of the waste in griff bins, you have to label it as OHC5, and usually they get thrown away sort of half full based on the time. So what we wanted to do was we wanted to put in place a safety assay to assess the safety of the compounds that we're making. So we in doing so, we put together a DOE where we were looking at what concentration of the PROTAC of interest can we actually use in our control. So Cassie, who put this together, ran her DOE. She optimized a number of different elements, but the one of interest to us was that you could reduce the PROTAC concentration used as our control from 500 nanomolar down to 5 nanomolar. And that 100-fold reduction in concentration that we used meant that the handling on the benchtop becomes so much easier because we've reduced that risk down. So when we went on to look at the, put together the assay and run it across 37 different compounds, we saw that in the top left, you can see CC885, which is our IMID, and you can see it's nanomolar potency. But we found that when you attach the same binder to a PROTAC, it reduces its potency in this safety assay. So we see over 100-fold reduction in potency in our safety assay. And on the right, we've got 2 different PROTACs with our in-house binder, and there's no activity against any of the 4 neosubstrates that we've checked there. So using this data, we were able to put this assay in place to test future PROTACs that we produce, but also to reduce the classification of our cereblon binding PROTACs, which meant that in the end, we can weight all of the compounds in the same hood, so we don't have to use glove boxes, which has led to an energy saving of 17.25 kilowatt hours over the year. We reduced the plastic needed to deal with these compounds by 6.75 kilograms. That comes from the double bagging and then the waste from the griff bins as well. And we also run this all in one experiment. We were able to do the setup for this, determine the optimal conditions in one experiment instead of what Cassie estimated to be 4. The second example I've got is a reduction in our protein that we're using. So this is for an IAP E3 ligase, and those are a family of E3 ligases which lead to ubiquitination and degradation of proteins. So the program which we were focusing on this assay for needed to target XIAP BER2. So each IAP has 3 BER domains, and you need selectivity for one of them over all of the others. So in the US, we had 6 assays set up for the 3 BER domains in XIAP and CIAP. So we wanted to transfer that to the UK. And as we were doing that, we noticed that there was 3 assays where the BER concentration was at 10 nanomolar, but then there was 3 where it was at 350 nanomolar for these legacy assays that are in place. So we wanted to employ a DOE to reduce the concentration of those 3 BER proteins down, including the one that we were interested in, which was XIAP BER2. So in one experiment, again, we were able to see that we could reduce the enzyme concentration for those 3 BER proteins from 350 nanomolar down to 50 nanomolar. And so we've saved 7-fold worth of protein there for 3 different proteins, whilst in a later experiment, we were able to see that we retained the plate statistics and also the potency didn't shift when we implemented the new conditions. And this is the slide that I'm not particularly happy with, because I couldn't find very accurate data for how much energy it takes to produce small amounts of recombinant protein. So I had to scale down from large production. So if there's any protein scientists in the audience who would laugh at 1.22 kilowatts hours of energy to make like 10 mgs of protein, I also don't think it's right. But if you've got a better number, then please let me know. And then my last example is around differentiated B cells. So some subsets of differentiated B cells are enriched in patients with autoimmune disease. And because of that, we wanted to do a chemogenomics screen where we could then identify the pathways and genes involved in the differentiation of those cells. In the literature, Tim found a set of conditions for media constituents to drive the differentiation. But when he attempted it based on what was in the literature, the data was quite poor. We saw poor cell viability. So we enlisted Synthace to put a DOE together, and we tested the different media constituents at a range of concentrations rather than just the fixed concentration that we did at first. So our aim was really to maximize the percentage of differentiated B cells, but also keep that cell viability up. I've included this table just to show what we did. So you've got the conditions that Tim ran initially in the middle, and then you can see that we basically did a buffer either side for each of the different reagents, keeping the cells per well the same. And the data was really good. So there were 3 key components that came out in the data. So 3 of them had an effect on cell viability, and then 1 had an effect on the percentage of differentiated B cells. And if you can see, there's one factor which is the same between both. So cytokine 3 had an effect on both the percentage of differentiated B cells, but then the cell viability as well. And the funny thing is that in the literature, it was one of the constituents that was played down. It wasn't one of the ones that people had mentioned as a key constituent. So in reality, I say this on the next slide, but if we hadn't have done a DOE, it probably would have taken ages to get to the conclusion that we did in the end. And so I've gone really quite granular on this just to make it stick out. But we think that it would have took at least 4 experiments instead of the 1 that it did, which means that we would have used 6 more microplates, 6 more flasks, 12 more Falcon tubes. We would have used 6 more pairs of gloves. There would have been energy use in terms of the incubators, the biosafety cabinets, the liquid handlers that were used in the process. So because we did that in 1 experiment, we eradicated all of that use. And the biggest thing, I don't know if anyone was here for in the tech theatre for Gareth Langley's talk yesterday, where he mentioned that the biggest savings with DOE are on those complicated experiments. I really haven't done justice how complicated this experiment was, because I've had to blind everything. But we don't know whether we would have actually got to the conclusion that we did in the end. And this is quite a long experiment to run, you know, it's labor intensive. So without the DOE, we might have just ended up canning this part of the project effectively. So that's a real big thing to note, that it's hard to measure. If you don't think you would have got to the conclusion in the end, it's hard to measure how much you would have wasted. But that's it for me. I'll pass back over to Ross.
Right, thank you very much, Harry. It's really interesting. So just a quick summary from me. I mean, hopefully we've shown you today how Synthace and DOE can ultimately empower day-to-day sustainability gains for scientists in the lab, how DOE can help you to reduce waste and gain some really great insights from the data and the experiments that you do in a way that is much more efficient. We've seen some both long-term, short-term improvements in terms of, you know, each assay that you do can be done in fewer runs and fewer experiments, but also those long-term benefits as well of, you know, needing less protein to run your assays, and that's going to compound over the years as you keep running that assay into the future. So just a quick point on some of the sustainability efforts that we've done both at GSK and at Synthace. So we're both part of the My Green Lab certification scheme. We're both looking at increasing the temperature of our cold storage freezers, and as well some nice sort of lab recycling schemes as well. So looking at disposing of that waste in a reasonable and sustainable way. So yeah, that's pretty much it from me today. I just wanted to say we do have a booth at Synthace. You can find us in B14. Want to hear more, whether that's about DOE, automation, the platform in general, do please come say hello. We'll be more than happy to chew your ear off about DOE. And also finally, Harry's nicely mentioned it already, but we did have a talk yesterday in the tech theatre from CRL. We've got a case study, so if you want to hear a little bit more about that, do come and say hello. With that, thank you very much, everybody. Welcome questions.